Good morning to everyone. Today we are going to discuss about Jablonski diagram and photophysical process. This belongs to Physical Chemistry 1 in 3rd BSc Chemistry. In Physical Chemistry 1 consists of 5 units. Unit 1 Group Theory and Photochemistry, Unit 2 Thermodynamics 2, Unit 3 Thermodynamics 3, Unit 4 Solution, Unit 5 is the phase rule. Now, I share my views in Jablonski diagram and photophysical process for photochemistry 1 for module 2. Now, let us see Jablonski diagram. Alexander Jablonski was a Polish physicist. He was known for the study of molecular absorbance and emission of light. He developed a written representation about consequences of applied photons. That scheme was referred as Jablonski diagram. A Jablonski diagram is a good starting point for the discussion of luminescence principle is simplified by Jablonski diagram. In the Jablonski diagram is basically an energy diagram arranged energy on a vertical axis and grouped horizontally by spin multiplicity. Now, radiative transitions are indicated arrows and radiative transitions by straight arrows. The vibrational ground states of each electronic states are indicated with thick lines. The higher vibrational states with thinner lines. The energy levels can be quantitatively denoted, but most of these diagrams use energy levels systematically. In the every column usually represents a specific spin multiplicity for a particular species. Column containing the electronic state in singlet and triplet state. In a singlet state is denoted as capital S and triplet state is denoted as capital T. So, column contains electronic state in singlet and triplet state. As electronic energy states increases, the difference in energy become continually less, eventually becoming a continuum that can be approached with classical mechanics. Additional, as the electronic energy level get closer together and overlap of vibrational energy level increases. Usually, only a portion of this vibrational energy states are represented due to the massive number of possible vibrations in a molecule. That the absorption of light in Jablonski diagram, the absorption is a phenomena. The absorption of a photon of a particular energy by the molecule result in migration of electron from lower energy state to higher energy state. This is indicated by straight arrow pointing up. Only certain wavelengths of lights are possible for absorption. They have energies that correspond to the energy difference between two different energy states of the particular molecule. The excited species can return to the ground state by losing all its excess energy by any one of the following process. It is non-radiative process or radiative process. First we see non-radiative process. In the non-radiative process involves transition from S2 to S1 and S3 to S2, that is singlet second state to singlet first state or singlet third state to singlet second state or T2 to T1 or T3 to T1, that is in a triplet state. It does not involve emission of only radiation. It only involves of emission of heat. There are three non-radiative processes: vibrational relaxation, internal conversion, intersystem crossing. First we see vibrational conversion. that the migration of electrons from higher energy state to the lower energy state by losing their absorbed energy, that is relaxation. This process involves the dispatch of energy from the molecule to the surroundings, and thus it cannot occur for isolated molecules. The timeline between the vibrational relaxation, the transition occurs from 10 to the power minus 12 to 10 to the power minus 10 seconds. This is indicated as a curved arrow between vibrational level. If relaxation occurs between vibrational levels from one higher electronic state to another lower electronic state, this phenomena is known as internal conversion. In this process, energy loses in the form of heat. It involves transition from S2 to S1, S3 to S2, or in a triplet state T2 to T1 or T3 to T2, which occurs when a vibrational state of an electronically excited state can couple to the vibrational state of the lower state. That another one non-radiative transition is the intersystem crossing. In the intersystem crossing, another path of the molecule may take in the dispatch of energy. This, where the electron changes spin multiplicity from the excited singlet state to the excited triplet state, indicated by a horizontal curved arrow from one column to the another column. This transition occurs from S2 to T2, S3 to T3, S1 to T1. That means that the singlet second state to triplet second state, or singlet third state to triplet third state. So, this is the very slowest process and also several orders of magnitude slower than fluorescence. It is radiational less transition between different spin states. Next we see radiative transition. In the radiative transition involves transition from S1 to S0 or T1 to T0. It involves emission transition. There are the two processes of radiative transition. One is fluorescence and another one is phosphorescence. First we see fluorescence. The emission of transition from S1 to S0, that is known as a fluorescence. That the observed between the first excited electron state to the ground state. That the migration of electrons from higher energy state to the lower energy state by emitting the photons. This is indicated as a straight line going down to the energy axis between electronic state. The allowed transition and also timeline between 10 to the power minus 9 to 10 to the power minus 7 seconds. It is not a very likely path of relaxation. And another one is the quenching of fluorescence. What is meant by quenching? If the excited molecule are deactivated and the fluorescence stops, the phenomena is called quenching. When the activated molecule undergo a change from a singlet excited state to triplet excited state, that is known as internal quenching. When the activated molecules collide with the other molecules or quenchers, which are the externally added species, and transfer their energy to those molecules, that is known as external quenching. Now, let us see the fluorescence, the other radiative transition. The emission of radiation from triplet to singlet, that is T1 to S0, that process is known as phosphorescence. It is forbidden transition. In the fluorescence, even after incident light radiation is cut off, there is emission of light for some time. It is also called fluorescence. The substance which shows fluorescence is called fluorescent substance. The name phosphorescence means glow in dark. So, now let us see photophysical process. The another chapter is the photophysical process. What is meant by photophysical process? That if the absorbed radiation is not used to cause a chemical change, it is re-emitted as light of longer wavelength. That is photophysical process. There are 4 types of photophysical process which can occur. That is fluorescence, phosphorescence, chemiluminescence and bioluminescence. First we see fluorescence. Fluorescence means in photophysical process certain molecules or atoms when exposed to light radiation of short wavelength. Short wavelength means high frequency. Emit light of longer wavelength. The process is called fluorescence. And the substance that exhibit fluorescence is called fluorescent substance. Fluorescence stops as soon as the incident radiation is cut off. The examples of the fluorescence. There are the many examples. The first one is the solution of quinine sulfate on exposure to visible light. It is a colorless, but it exhibits UV light that it shows as a blue color. And another one is a solution of chlorophyll in ether, shows blood red fluorescence. And another one is the minerals in UV exposure. What it is occurs in this process? When a molecule absorbs high energy radiation, it is excited to higher energy state. Then it emits excess energy through several transitions to the ground state. Thus, the excited molecule emits light of longer frequency. The color of fluorescence depends on the wavelength of light emitted. The next one is phosphorescence. Phosphorescence, when a substance absorbs radiation of high frequency and emits light even after the incident radiation is cut off. The process is called phosphorescence. The substance which shows phosphorescent substance. Examples are sulfates of calcium, barium and strontium exhibits phosphorescence. When the fluorescence in boric acid shows phosphorescence in the blue region at 5700 angstrom wavelength. As in fluorescence, a molecule absorbs light radiation and gets excited. While returning to the ground state, it emits light energy of longer wavelength. In doing so, the excited molecule passes from one series of electronic states to another and gets trapped. This shows the emission of light which persists even after the removal of light source. Thus, phosphorescence could be designated as delayed fluorescence. The next one is the chemiluminescence. Some chemical reactions are accompanied by the emission of visible light at ordinary temperature. The emission of light as a result of chemical action is known as chemiluminescence. This reaction is referred to as chemiluminescent reaction. The light emitted in the chemiluminescent reaction is also called cold light because it is produced at ordinary temperature. There is one example, the oxidation of 5-aminophthalic acid hydrazide, that is known as luminol, by hydrogen peroxide in alkyl solution producing bright green light. In the chemiluminescent reaction, the energy released in the reaction makes the product molecule electronically excited. The excited molecule then give up its excess energy as visible light while returning to ground state. Another one is the bioluminescence. It is a form of chemiluminescence. Bioluminescence is the protection and emission of light by living organism. Bioluminescence occurs widely in marine vertebrata and invertebrata, as well as some fungi, microorganisms, including some bioluminescent bacteria and arthropods such as fireflies. In the fireflies, the aerial oxidation of luciferin in the presence of enzyme luciferase. Luciferin is a protein. In the protein luciferin is at the oxidation with in the presence of luciferase enzyme to give us a oxyluciferin plus light energy. That light energy is released by a chemical reaction. The reaction involves a light emitting pigment, the luciferin and its enzyme. Because of the diversity of luciferin and luciferase combination, there are very few commodities in the chemical mechanism. The firefly, luciferin and luciferase requires magnesium and ATP and produces carbon dioxide, and also adenosine monophosphate and pyrophosphate as waste products. Other cofactors may be required such as calcium or magnesium ions and ATP for the firefly luciferase. In the case of jellyfishes, they mostly produce luciferin in their phytocyte cells. The same reaction occurs in the jellyfishes also. When an ion such as calcium is present, the process occurs that emit light. These photoproteins are found in the photocyte cells of bioluminescent jellyfish. So, an organism must constantly introduce more luciferin into its system to glow constantly. Some animals get it from their diet, while others make their own. Most luminescent jellyfish occurs in the deep sea. In the deep sea, bioluminescent jellyfish live often in the total darkness. So, they come up to the ocean surface at night to feed a plankton and the other organisms by creating a disturbance in the water, which attracts their food close to them. Now, we are learned about the Jablonski diagram and the transitions of the Jablonski diagram in photophysical process, that is such as fluorescence, phosphorescence and chemiluminescence. Thank you.